This is the first part of the second lecture. We're going to talk about the visual system. For human beings, vision is considered to be the dominant sensory modality. And of course, this is assuming a human being with a fully functioning sensory system. Now, this is generally considered to be the case for two factors. One, more of the cortex, the cerebral cortex, is devoted to visual processing than the processing of any other sensory modality. Further, when we experience conflicting sensory input, we oftentimes will give priority to visual cues. This is how ventriloquism works. So in case you don't know, ventriloquism is when there is some performer who has a doll and they're making the mouth of the doll move and they're actually speaking, but it seems to us as if the doll is the one doing the speaking. The reason for this is even though we hear, and when we talk about hearing, we'll see that we have very good location of where sound is coming from, but we see also that the mouth of the doll is moving and there is a disconnect in that interpretation, sound coming from one place and the visual cues of mouth moving in somewhere else. Yet our brain will override the auditory cues and use the visual cues in a primary sense of locating where the sound is coming from, and this is why we see the sound and hear it then ultimately coming from the doll. And that is a unifying process which we will talk about briefly in this lecture, but as we move through this course, we'll talk much more about it. The sensory tool we use to detect any kind of visual stimulus is the eye. It works by ultimately detecting reflective light. Please, I'm sorry if this is a silly thing to say, but you'd be surprised how common, unfortunately, this is. The eyes don't work by projecting anything out into the world, not even any kind of eye beams that somehow perceive things. Nothing goes from your eye out into the world. It's all about how light in our environment reflects off of the objects around us, and thus some of that reflected light goes into our eye, and this is how we perceive the world. Now, light passes through both the cornea and the lens, and both of these are involved in focusing light. The cornea specifically focuses light onto the lens, and then the lens itself will focus light onto the back of the retina, specifically oftentimes trying to get it to be focused onto the fovea. Now, just an extra piece of knowledge here for you. While I know organ donation can be a touchy subject for many people, please note that if you are, cornea transplants are one of those things that are one of the most sought after transplants for people. The light then falls or is focused onto the retina itself, and the retina is where our photoreceptors are located, and we will talk a bunch more about these in a couple slides after I talk a little bit about light. Now, also you can see here for your viewing pleasure, a picture of how light is flowing through the cornea and the lens, and it is going onto the back of the fovea. A lot of times you're not going to see such a broad image cast on the fovea, it is more specific. And another part of just how optics work is that the image cast on the retina is actually upside down. So some background information about light. Yes, this is not a physics class, but it is important to know because ultimately this is what our sensory apparatus in our eyes is detecting. So when we talk about light, we're actually talking about a very small portion of what is otherwise known as the electromagnetic spectrum. And this includes all kinds of different classes of waves. Gamma rays, X-rays, which we use for actual X-rays, and ultraviolet light, which is what gets you your tan or can give you skin cancer. These are all on one side of the visible spectrum, and on the other side of that spectrum is the infrared, microwave, and radio wave frequencies, which infrared is the heat that we get off from sitting next to a fire or a heater, or what radiates from the sidewalk after it's been in the sun all day. Microwaves, many of you are familiar with. These are the waves that we use to heat up our food in microwaves. And then radio waves also are very, well, actually, they might not be that familiar anymore. These are when you use a radio to listen to all kinds of things that are broadcast around the world, and it's what most people use to listen to entertainment before satellite networks and internet was available wherever you went. For the sake of this class, light is going to be considered a wave. Some physicists will consider it a partial between a wave and a particle, but I'm not going to get into that. It has three main characteristics that you should be aware of. The first is wavelength, and this refers to the distance between two peaks of the wave. It could be the distance between two subsequent troughs also, that's what the dips are called. And a way to visualize this would be to think about the swells on an ocean, and this would be the distance between the topmost parts of two of those swells or humps in the water. The amplitude is how high each of those wave peaks reaches, and this could just be that how high or low those ocean swells go. There is a much greater amplitude to ocean swells in a storm than when the ocean is calm. By that, it just means that the swells go much higher and lower when there is a storm versus when it is calm ocean, they are relatively small. Frequency refers to how many complete wavelengths could pass through a single point each second. It is important to note that wavelength and frequency are inversely correlated, so as wavelength tends to get very high, frequencies will tend to be lower. And it is measured in what is called hertz, and hertz is just how many cycles per second. So if you were to tap your finger 10 times a second on your table, you would be tapping your finger at a frequency of 10 hertz. Now, the entire electromagnetic spectrum, like how I just told you, going from X-rays, which can let us see our bones, to visible light, which lets us see everything, to microwaves, ultimately, which let us heat up our food, or radio, which lets us hear things through the air, basically. All of them are part of the same thing. Now, a single unit of this energy, no matter where you go on the spectrum, is called a photon. All photons, basically, at least in vacuum, zip around at the very speedy rate of 300 million meters per second. I'm sorry I'm not saying this in miles and stuff, but it's, I guess, I preferred scientific units at least, and that's what I grew up with. Now, just even then, for reference at how staggeringly huge the universe really is, even at that speed, right, which is an insanely fast speed, it still takes over 8 minutes for light to get here from the sun, which just goes to show you how far away it is. Now, you'll find gamma rays on the shortest wavelength side of the spectrum, and these are generally of the order of 1 times 10 to the power negative 12 meters, or here I've given you the decimal as well, or a thousandth of a nanometer, sometimes called a picometer. On the complete other side, you have radio, and radio has enormous wavelengths, sometimes up to a kilometer or greater. This is why we use it for radio broadcasts, because it covers such a wide range as it's moving through the atmosphere. Now, visible light is between 400 and 750 nanometers. Just for reference, 1 nanometer is 10 to the power negative 9 meters. Approximately 1 billion nanometers fit in 1 meter. Excuse me. I'm just going to turn on the dot here. And so here you can see how small a portion is actually represented by visible light, or just white light in general, but you could split it apart with a prism. And the colors, as we'll talk about in a second, are just ultimately our way of indexing different frequencies or wavelengths of light within that spectrum. As wavelengths are closer to 700 to 750 nanometers, we tend to see that as red. And then as you move down the wavelength towards about 400 nanometers, we begin to see that as blues and purples. Also, it is a, I guess, fun fact to know that not all creatures see exactly the same spectrum. They see different parts of it. Seeing red actually is a relatively new adaptation, which is in, some weird fish actually, but also in mostly the apes, because some people assume it was to allow them to differentiate the difference between the greens of the trees and the reds of a lot of the fruits that you found in jungles. Some creatures cannot actually even see red light. Like a fun one that you could experiment with are ants. If you take a red light in the dark and you shine it on them, they will not react at all. You can spy on them to your heart's content without ever giving them a clue that you're there. Whereas if you were to shine a direct white, like a bike light for example, onto them, they will all freak out at night. So going back to photoreceptors, photoreceptors are specialized neural cells that we find in the retina of the eyes, and they're able to detect photons in the range of 400 to 750 nanometers. This is why we call that part of the spectrum visible light, because, well, we can see it, and many other things can see it too. Now, it's also, I think, an interesting quirk of the world that photosynthesis actually occurs in this range also, mostly with the reds and blue lights. We see the green of plants because that is the light that is often not used in photosynthesis, and thus is it reflected back from the plants. The more you know, right? Okay, there are two specific types of photoreceptors in the retina. They're the rods. These are photoreceptors that are sensitive across pretty much all light conditions, and they are also sensitive in very low light conditions, which the cones are not. They are, however, not able to discern the difference between different wavelengths and are thus not able to give us any information on color. The cones, on the other hand, allow us to see color because we have three different kinds, and each different kind is attuned to a different wavelength of visible light. Now, the way they allow us to discern different colors, especially the huge gradient of hues that we're able to see when we only have three of them, is that they're firing at different rates. And we'll talk about this more as we go through this lecture of receptive fields. But so there are some that are attuned to the lowest wavelength, so the blues, the 400 nanometer range. And so if a color is coming into our eye that is blue, those are going to be the cells that are firing the most often. They're most excited. But that does not mean that the other sets are not firing too. They'll just be firing at a much lower rate. And it is an aggregation and comparison of the firing rates of all three types of cones that allow us to perceive colors. And of course, this ratio between firing of all three will change depending on what color you're looking at. Some people have certain types of color blindness, and this can result from a lack of cone being represented in their eyes. This is also why the red-green color blindness is the most common, because phylogenetically, those cones are the most new. Now, cones also allow us to make out fine details, which our rods are not so good at. Speaking of fine details, this is referred to as acuity. Now, these are distributed across the retina, but not in an equal fashion. Oh, move this out of the way. So you're going to find most cones in the fovea, which is, remember, where your lens is trying to focus light, which makes sense because your cones are also what allow you to see the most detail, and so it makes sense that your lens is always going to try and focus light onto the fovea. Now, rods are located throughout pretty much the rest of the retina, not to say that there aren't some in the fovea itself, but much less so in the rest of your eye. Now, here you can sort of see a distribution of how that looks. The orange graph is representing the number of cones, and the purple is representing how many rods, and this is how many per square millimeter. This zero line in the middle represents the distance from the fovea. So zero would be right in the middle of the fovea. And here you can see there are tons and tons of cones and almost no rods whatsoever. But as you move away from the retina in either direction, the number of cones drops off pretty dramatically. Not that there are none, but there are very few. Now, rods, on the other hand, follow the opposite pattern. As you move away from the retina, you see a drastic increase, but eventually they too will fall off. This is to imagine if you were to go right up next to your lens from the inside of your eye, there's no need to have photoreceptors there because there's no way you can get light there, because you can't have light go through your lens and then curve around. For the most part, light travels in straight lines. Now, this is an electron microscope's picture of the rods and cones. They don't actually have this kind of coloring, but they are named after their namesakes, where this little oblong shaped thing is the cone, and these little stick things that look like chip puffs are the rods. Now, oh, no, we won't even go into that. Never mind. Just a reminder about parallel processing. So we've already discussed this, but this refers to the processing that is done in parallel or at the same time. This is contrasted with serial processing, which is processing one thing at a time in a sort of linear sequence. I do one thing, I do another thing, I do another thing, and then I do the last thing. That would be serial processing. If I was somehow able to do 4 or 5 things at the same time, that would be parallel processing. Now, a note here, this fact that the brain does most of what it does in a parallel way does not mean that we can multitask. Rather, the whole brain is always working on many, many small tasks simultaneously, and that gives us our moment-to-moment experience. Basically, your whole brain does you every single moment. So please don't text and drive, because you cannot actually do both things. All you're doing is switching back and forth pretty fast, but at the end of the day, while you're texting, you are not actually involved in driving, which is horribly problematic because most cars weigh over 1,000 kilograms, and that can mash a lot of things. It is ultimately not cool to be able to do this, no matter how much you can lean back and make a cool pose. And if something is really, really that important, just pull over and enjoy the important thing. Now, that's just me saying it. At the end of the day, you are free to do as you would like. Before going further, there are a couple of other cells in the retina to be aware of. There are 2 additional types. One is called bipolar cells, and the other type are called ganglion cells. Now, information flows from the photoreceptors through the bipolar cells into the ganglion cells, which then ultimately transmit information out of the eye into the thalamus itself, specifically what is called the lateral geniculate nucleus, and from there into the back or area V1 of the occipital cortex. Don't worry, we'll talk about those regions and names a bit more as we move on. Before going there though, it is important to note that the processing of visual stimuli actually occurs already in the eye itself before any signals have even left the eye itself. Now, this is referred to as lateral inhibition, which is this processing stage, and it's conducted largely by the bipolar cells. Now, lateral inhibition allows us to see edges more clearly. It basically highlights them and gives additional contrast. Here is an illusion to demonstrate this, and this illusion is often called the Mach band illusion. Now, while many of you can probably see that there are lots of different bars through here of varying shades of gray, what really makes this, or what shows this process, is if you were to identify one of these bars, let's say this one, and you compare this side of the bar to this side of the bar, you look closely. I would recommend doing this maybe in your textbook or on a large screen. You'll notice that the side of the bar that's closer to the light one is actually a little bit darker than the same bar's side that's closer to the darker one. And this is an illusion created by the process of lateral inhibition, right? It's ultimately serving to enhance that edge or boundary between these 2 different shaded gray bars. Now, how does it work? Well, please remember before we jump into this, because this can sometimes get people all kinds of lost, but hopefully this will make at least some degree of sense. Now, cells fire at specific rates. Remember, this is how we measure how excited or inhibited they are, not about the magnitude at which they fire. Remember the all or none law. So it's basically all about how many times per second are they firing, or in this case they refer to it as spikes, but spikes and fires are ultimately interchangeable. Now, here you have the photoreceptors themselves. These little green lines are recepting or are representing bipolar cells, and then these extensions are the ganglion cells moving on down to the brain. Here we see that 2 stimuli are being presented to the eye itself. You could think of this as 2 of the bars represented in that Mach band illusion, one being gray and one being bright, or just a lighter shade of gray if you wanted. The reason it says intense stimulation here and moderate stimulation there is because oftentimes bright stimuli are detected more strongly, and this just means that the photoreceptors underneath this bright light are firing at a higher rate than photoreceptors that are under a softer or dimmer light. Now, as this goes on, you see that there are different numbers of spikes per second. We see that these two here have about 1,000 spikes per second. This one has more than them for some reason at 1,100. This is at 80, which is actually a little bit less than these two over here. So how is that working? Well, this is because of the function of bipolar cells. So while this photoreceptor is getting activated, it's firing at several, or this 1,000 spikes per second, let's say, it is not only activating the ganglion cell, but it is also activating these two bipolar cells. And these two bipolar cells will ultimately inhibit what they're connected to as much as they are themselves excited. So basically what bipolar cells do is as they themselves get excited, they try and inhibit whichever cells are next to them. And this is spread out through the entire retina itself. So here you have this one that's getting 1,000 activations per second, let's say, or maybe a little bit more to then deal with this inhibition. And then this cell and this cell are receiving the same amount of activation. So they are inhibiting this neuron also, as much as these are inhibiting all the others. And this just sort of cascades down, and this is why each of them is 1,000. This one, however, is a little bit different. This is a neuron that is brightly stimulated, firing at a very high frequency. It's being inhibited strongly from this side, but from this side, it's not being inhibited as strongly, because this cell is only firing at about 80, let's say 100 if you wanted to go beyond the inhibition to be specific. Here, the inhibition coming through this side of the bipolar cell is much less. So overall, this ganglion cell is receiving less amounts of aggregate inhibition than, say, these two. This is why this has a higher rate of firing than these two over here, and continuing over this way until a different edge is detected. The opposite is happening on this side. Here, these are all being stimulated at relatively low rates. They are also inhibiting each other at relatively low rates because they don't have that much activation flowing from the photoreceptors into the bipolar cells themselves. That story changes at this one right here, where you have low activation coming in, a low degree of inhibition coming from this side, but since this one is strongly activated, it is also strongly inhibiting this one from this side, which is what results in this having a lower number or a smaller activation than these two over here. And because this is now slightly higher, we perceive this as a little bit brighter than all of these, and because this is a little bit lower than these two, we perceive this as a little bit darker. This is why we perceive the side closer to one of the dark sides as being lighter than the rest of it, and the dark side closer to the lighter sides as being a little bit darker than the rest of that shade. Now, I would highly recommend going back to the mock bands or pull it up on your computer and look at it and try and understand how those contrasts are happening in regard to this neural structure. Moving on to receptive fields. So a receptive field refers to the specific type of stimulus a specific neuron is attuned or receptive to. So we already, I'll get to that example, sorry. Generally speaking, the closer the actual stimulus is to the preferred stimulus of that neuron, the higher its firing rate will be. So it's not an all or nothing business here. It will be as close as it is to the desired input of the neuron will fire maximally there. It will fire repeatedly many times per second. But as you move away from this, it will fire at less and less speed. There's a visual example on the next slide for this in case that's a little bit unclear. It's also very important to know that while an entire cluster of neurons, let's say, might have the same type of receptive field, they're interested in seeing horizontal lines, let's say, they generally will be subdivided further based on what part of the visual field itself that they are receptive to. So you could have some that are receptive only to the top right corner of your visual field, other to the middle left, and still more for the bottom left corner, and etc., as you got through the entirety of the visual field. Now, an example of receptive fields before we get into our line detectors is that each type of cone is receptive to a specific wavelength. It doesn't mean that they don't fire for the other wavelengths, but they fire most for the wavelength they prefer and less so as you move away from the wavelengths that they are the preferred ones for those cones or photoreceptors. And of course, they don't actually like it in the sense that human beings like things. We just mean preferred in the sense that that elicits a maximal response from that photoreceptor. Here is an example of a common type of receptive field, and these are neurons that are specifically dedicated to seeing lines, or another way of referring to them would be line detectors. Now, the visual cortex itself can be subdivided into a bunch of areas. We'll talk about that on the next slide. And the one specifically we're looking at here for line type detectors is area V1, which is at the backmost part of the occipital cortex and is actually what receives information from the eyes via the thalamus. Now, these lines here are to represent the orientation of lines presented to this neuron. Well, technically to the eye, and then it goes to this part of the cortex that has this specific neuron in it. But here is to show the type of firing rates that are present in this neuron. So this is obviously a neuron that is attuned to vertical lines. And you see this by all of these little hash marks, which represent the firing rate of this neuron. As you move the orientation away from vertical, it starts to fire at a lower and lower frequency. And you can see once it's maximally away from its preferred orientation, it fires seldom, if at all. Now, we have neurons whose receptive fields come for almost pretty much any type of orientation you can imagine. So it's not just vertical and horizontal lines, but it is diagonal lines that it will fire most to. And let's say hypothetically we were interested in a diagonal line neuron, it would fire maximally here and less so here. So basically you would have this image on both sides. To get this kind of inhibition, you would need basically another diagonal, but that was offset by 90 degrees. So looking kind of like that. And because we have so many of these, it basically allows us to perceive all different types of lines. Now, there are several areas of the visual cortex that are good to know about at least, and these are subdivided based on what types of neurons and what types of receptive fields they have. So we already said about area V1. Well, before we jump into that, so here's the retina. The retina is passing information via the ganglion cells into the thalamus. This LGN stands for the lateral geniculate nucleus. There are many nuclei in the thalamus, and many of them are subdivided across what modalities of sense are coming in, along with some additional functions. Anyhow, information passes from the lateral geniculate nucleus into area V1, and from there it propagates outwards through the brain. Now, area V1, as I mentioned, contains most of the line detectors. Here you can go on then, let's say, to area V2, which is oftentimes responsive to angles. So let's say you had two different sets of line detectors that are picking up two different lines that are 90 degrees from each other. You will have specific neurons with receptive fields in area V2 that are receptive, maximally to 90-degree angles, just like you will have some for 30-degree angles and 42-degree angles and so on and so forth. Moving on, area V3 is oftentimes receptive to different types of shapes, triangles, circles, hexagons, and so on. Area V4 has a combination of receptive fields looking at shapes and color. And then areas MT and PO are both responsive to motion, even though arguments have been put forward recently to say they look at different types or are receptive to different types of motion. Now, one other thing to be very careful of, or at least to just keep in your mind for most of these lectures really, is notice that there are these double-headed arrows that are going between most of these regions. That is to indicate that they are interconnected and they are sending information both ways. This is to illustrate that ultimately the Neurons that are receptive to angles are influencing the neurons that are receptive to lines, just as the neurons that are receptive to lines are influencing neurons that are receptive to angles, which are also in turn influencing, say, neurons that are receptive to motion, which are in turn again influencing neurons that are receptive to angles. This interconnectivity is a hallmark of how the brain is organized. Now, just an extra fun fact, I guess, for you is that area V4, which is oftentimes associated with the processing and perception of color, for people who have synesthesia, this is when they're reading words, for example, they also experience color. They have a specific track of white matter, it's a neuronal connection, going from their language centers to this region. It is actually detectable. And this is one of the reasons why they experience color when they are reading or thinking about words. It's not that they see letters in blue or green, but rather they have the experience of those colors as they're having the experiences of those words. Moving beyond that visual cortex itself, so there are two areas in the brain that are highly connected with the visual cortex itself and are also involved in the processing of visual stimuli. The first is in the parietal cortex, that's up here, and this is oftentimes referred to as the where pathway. This just means that information here is used to establish where in space various items in the visual field are located. The other pathway is called the what pathway and travels down to the temporal cortex, and this is largely involved in determining what specific objects are, or understanding what content they have, or what labels to give them. Now, specific damage here will result in specific impairments. So if you suffer damage to this region of the posterior parietal cortex, you will end up having trouble locating things in space accurately. You would have great trouble actually reaching out and grasping something, but you would still be able to look at images and name what they were without any problem. If you alternatively suffer damage to the temporal cortex without having damage in the parietal cortex here, you would be able to reach and pick things up without any problem at all, but looking at them, you would not be able to say what exactly they were. This is also true for damage to other parts of the visual cortex. For example, if you suffer damage acutely to area V4, you will lose the ability to perceive color. Now, this is different than color blindness that happens because of, say, a lack of rods in the eye. When you have damage here, the ability of perceiving color is gone in its entirety. Not only will you not see color when you look out at the world, but you will no longer be able to imagine colors or dream in colors. Literally, color will be gone from your experience. Now, we've talked a lot about how all these different regions seem to all be working on different parts of the problem at the same time and in different parts of the brain. And so it raises the question of how then do we have this unified visual experience? How is it that the brain is then able to reintegrate all of these different parts? And this is referred to as the binding problem. And while it has given a lot of theorists and scientists a lot of trouble over the years, there are currently three mechanisms by which this is thought to occur. The first has to do with spatial maps. So remember I said that not only are neurons receptive to some specific feature of a stimulus, but they're also only receptive in certain areas of the visual field. Some in the rightmost corner, some in the leftmost corner, some at the bottommost corner, and so on and so forth. If all the different regions are creating these types of spatial maps, those maps can then ultimately be overlaid upon one another, and it will allow the different features to be correctly positioned in space. A way to imagine this with a real-world analogy would be to imagine a map that was made up of multiple layers of trace paper. So your first one contained just the continents themselves, your second layer of trace paper considered the sovereign nation boundaries, the third layer considered where all the cities were, the fourth layer had all of the highways interconnecting these cities, the fifth layer had where forests were located, and so on and so forth. By putting these maps on top of each other, you could build a very rich understanding of what the continents actually were like. In the same way, it's thought that the brain reintegrates much of this information by overlaying and associating all of these spatial maps. Another component in allowing the brain to know what parts or what features go together with what specific stimuli is through something called neural synchrony. And this refers to different ultimately feature detectors, which are just neurons with specific receptive fields, firing in harmony or synchrony when two or more features are present in an object. Imagine, for example, that you were looking at a computer screen and you were seeing a red square moving from left to right. You would have activation, of course, in the lower areas as well, but for this example, let's imagine area V3 is activated because you're seeing a square, so parts of that area, along with parts of V4, which are also interpreting the square along with it being red, and parts of the MT or V5, which are now also firing in response to this object moving. Interestingly enough, they're neurons in MT that will fire only if something goes left to right, or from right to left, or diagonally from up to down, or it's also very specific. Now, these three different areas will all have clusters of neurons, and since they're all responding to the same type of image, they will all be firing in a certain type of synchrony. And this is thought to be one of the ways in which the brain knows that multiple different activities belong together. This is actually thought to be something that is sort of across the entirety of the brain, and how it is able to modulate all types of various complex thoughts and keep them all together as well. Finally, there's attention, and attention is quite fundamental to the binding problem, and we'll talk a lot about attention when we get to the chapter specifically on it, but it does appear to be essential for this binding problem. We know this through experiments on confabulation errors, which are experiments in which when you overload the attention of a participant, they will be able to successfully remember what objects and stimuli were presented to them, but they will mix up the features that go with them. This is an example of this would be if you were to show a red H and a blue T, it could be that on recall the participant will think the T was in fact red and that the H was blue, and they will mix up the features even though they can correctly remember that they saw an H and a T. One last thing on attention and confabulation errors. So I already mentioned what confabulation errors are, but I highly recommend not watching Netflix and using your phone when you're studying. You're essentially dividing your attention and overloading that attentional mechanisms, and while you might remember what you're learning, you're much more likely to mix up the specifics you learn about each concept. This is something most of us are unfortunately familiar with. So just try not to do it. When you're focused on studying, just focus on studying. And when you're just chilling, just focus on chilling. That being said, most of you are legally adults, and so please make your own choices.